Good afternoon, and welcome to the USDA Human Nutrition Research Center on Aging's weekly seminar series. Before I introduce today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. And I encourage you to add your questions as they arise during the presentation. Today, I have the pleasure of introducing our speaker, Dr. Jesus Gil. Dr. Gil is a professor at Imperial College London, where he is a member of the Department of Molecular Sciences and the Institute of Clinical Sciences. He also leads the Cell Proliferation Group at the Medical Research Council's Laboratory of Medical Sciences at Imperial College. His main research interest is to understand how cell senescence is regulated. His laboratory studies the molecular pathways regulating senescence and the senescence-associated secretory phenotype and their pathological implications. The ultimate goal of his research is to exploit the molecular mechanisms controlling senescence for therapeutic benefit. Today, Dr. Gil will present his research findings in a presentation titled Strategies or Cellular Senescence as a Therapeutic Target. And now let me turn the microphone over to Dr. Gil.
Thank you very much. So thanks for the invitation to the online seminar. And I'm going to talk about senescence and how we can use senescence as a therapeutic target. So senescence is a cellular stress response that is activated by both intrinsic stresses such as oncogenes or replicative stress, and also extrinsic stresses or insults such as irradiation or chemotherapeutic drugs. In response to all of those, cells undergo a number of phenotypic changes. So the most relevant or defining of this is this stable cell cycle arrest that is associated with senescence, but senescence cells change the morphology, remodel the chromatin, reprogram their metabolism, increase the number of lysosomes, and that can be used because there is an associated increase in beta-galactosidase activity to detect senescent cells. Importantly, also are resistant to apoptosis and secrete a combination of immunomodulatory factors that we refer as the senescence-associated secretory phenotype, or SASP. So senescent cells is a complex phenotype, so it's a multifaceted process. So perhaps the two more important parts on one hand is this stable cell cycle arrest that is controlled through damage and epigenetic activation that achieves the activation of the p53 and the p16RB tumor suppressor pathways, and eventually it finishes in the stable repression of E2F-dependent genes. And these E2F-dependent genes are necessary for moving into the cell cycle. On the other hand, so one of the other parts of the senescent phenotype that is very relevant for many of the effects of senescent cells is the SASP. So this secretory phenotype is this combination of immunomodulatory inflammatory factors secreted by senescent cells. And at the mechanistic level, this is controlled epigenetically by, for example, remodeling of super enhancers by BRD4, and transcriptionally by NF-kappa B and CBP. And as a result, there is a strong induction of pro-inflammatory factors and immune modulators. So altogether, the different aspects of senescence come into playing different effects in disease and in normal physiology. For example, in the context of cancer, we know that senescence is itself a protective response that limits cancer initiation and also dictates some of the responses to therapy. And this is both because of the stable cell cycle arrest. So this stable cell cycle arrest is an intrinsic response that makes that cells that, for example, have oncogenic mutations, instead of continue proliferating, become arrested. And here we have in this slide, so a section of the lung of a model of lung cancer, or mouse model of lung cancer. In blue, we have cells that are senescent, and this is basically an adenoma, a premalignant lesion of this lung cancer. And here in the bottom, there is like an adenocarcinoma, a more advanced lesion of this lung cancer. And we see that it's devoid of senescent cells. And part of what this indicates is that by inducing senescence, so we inhibit the progression to a more malignant state, and only when we have mutation that disable senescence allow the progression. On top of this stable cell cycle arrest, the SASP, through their immunomodulatory functions, is also relevant for these, in this case, protective effects against cancer. And for example, oncogenic activation, we saw that results in senescence, produce a SASP, not only this intrinsic growth arrest, but also the SASP. And the SASP is able to attract immune cells that will, in a extrinsic way, eliminate these senescent cells and avoid that potentially they can mutate and give rise to tumors. So senescence is protective, but paradoxically, senescence is also involved in aging and many age-related diseases. And this is just a list of diseases in which we know that senescent cells are present. And this is not just cancer, fibrosis, atherosclerosis, neurodegenerative disease, ophthalmological disease, and many, many others. And paradoxically, senescence there plays a negative role. And this was a puzzle for the senescence field for many years, these potential benefits of senescence as a way to avoid cancer progression, even avoid fibrosis. But on the other hand, this accumulation of senescent cells during aging and disease and their contribution to these detrimental phenotypes. Today, we have a model that seems to integrate these paradoxical effects. And for example, if we have a tissue in which we have like some damage in the tissue, one of the responses that can be activated is senescence. And what senescence tries to avoid is that cells that are damaged or cancerous proliferate and replicate in the tissue, replicating this damage that they have with them. And senescence achieves that through this intrinsic cell cycle arrest. On the other hand, senescent cells start to produce the SASP, and that in normal circumstances triggers an immune recruitment and will clear these senescent cells. So there are both intrinsic cell cycle arrest and extrinsic recruitment of the immune system to eliminate these senescence damage or cancerous cells. And in a normal tissue, the tissue will regenerate, and senescence is a net positive response. So it contributes to normal development, to normal tissue homeostasis, including proper wound healing, is tumor suppressive, and even limits fibrosis. The problem arises when this cycle of senescent cells induction and elimination does not go like depicted here, and senescent cells accumulate. And senescent cells can accumulate because there is damage in the tissue that is chronic, because there is like an immune system that is not effective in eliminating the senescent cells, and that might happen when you get old, or because there are active immune evasion mechanisms that senescent cells activate to try to avoid their elimination. And then is when senescence is negative. And one of the questions that we still have not answered properly is why is this accumulation of senescent cells has so many negative effects. And part of the current hypothesis, or the best explanation that we have, is because of the SASP. And this goes down to many years ago when we saw that, for example, the SASP of senescent cells was able to in an autocrine fashion, reinforce senescence, and in a paracrine fashion, induce senescence in other cell types. And this is an example where we have an papilloma that is senescence, a premalignant lesion of the skin. And we have this mark in red, but also mark in green here that is marking either P16 or P21, two senescence effectors. And what we can see is that when we look into green is marking for the keratin 5, and red is marking for the senescence markers. And when we look outside of the tumor, what we can see is that in the cells that are proximal to it, there are some that have markers of senescence being either P16 in the epidermis or P21. And this induction of senescence in a paracrine fashion is one of the ways in which the SASP can basically cause tissue disruption of the tissue homeostasis. And we believe that that's maybe one of the main reasons why having very few senescent cells is enough to disrupt tissue homeostasis and cause some negative effects in many diseases. And this is basically a summary. This aberrant accumulation of senescent cells is what trigger negative effects that are mediated by the SASP, by this chronic inflammation that basically disrupt tissue function, contribute to age-related pathologies, even paradoxically to tumorigenesis. And in this way, we can somehow put together these positive and negative effects of senescence. This normal cycle of elimination of senescent cells where senescence is good, and these negative effects associated with the accumulation of senescent cells. And obviously, because senescent cells are present in so many diseases and accumulate during aging, there is a need or a research in the field to try to see what we can do to try to mitigate these negative effects of senescent cells accumulating. We can do this in different ways, and I'm going to show you briefly some of the ways in which my lab has tried to identify ways in which we can target senescence. And this might go for a start, reducing the induction of senescence, so blunting the senescent response. Eventually, we saw that the SASP through chronic inflammation is what is causing these negative effects, and we could also try to inhibit this SASP that cause chronic inflammation and result in some of the detrimental effects of senescence. And the other thing that we could do is to potentiate the immune system in order to eliminate these senescent cells that accumulate aberrantly. And I'm going to show you some examples of this and the way that our lab tries to investigate and identify new ways to target senescence, and in parallel, understanding how these senescence mechanisms work, is by using phenotypic screenings. And typically what we will do is we have different models of senescence, either of senescence in normal or cancer cells, driven by oncogenes, by chemotherapy, by replicative senescence, in different cell types, fibroblast or epithelial, and also models in which we check how senescent cells interact with the immune system. A model that we use quite a bit and that I will show you later on in the talk is this model of oncogene-induced senescence, where we have fibroblast expressing a chimeric function, a chimeric protein of fusion of ER and RAS that can be activated when we add tamoxifen, the ligand of the estrogen receptor, and then it triggers senescence. And when we do that, senescent cells will basically start proliferating. Stop proliferating. They will start expressing P16 and P21, the cyclin-dependent kinase inhibitors. We will see DNA damage. We'll see reactive oxygen species, and we will see also the activation of P53 or the induction, the expression of the SASP. In this case, this is the intracellular expression of IL-6 that is being produced to be secreted. And with these models of senescence, we will combine them with libraries, being either genetic or drug libraries, where we can basically interrogate genes or drugs that are going to interfere with senescence phenotypes. And we do all types of screens, both pool screens, in which we check what happened in a population of cells, and we will just use RNA-seq to try to see for enrichment or depletion of CRISPR or shRNA vectors. But we also use a lot these high-throughput microscopy-based assays, where we can monitor in 96 well plates what is happening when we add different drugs or different siRNAs to senescent cells. And we look for different senescence phenotypes, and I will talk about some of these during the talk. So for example, one of the ways that I mentioned that we can try to target senescence is by blunting the induction of senescence. And we did that in our model of oncogene-induced senescence. And also we did it in another model of telomere uncapping, where we can inducibly express a dominant negative version of one of the telomere of the proteins that's basically cap the telomeres, so TRF2 is part of the cell-tearing complex. And this dominant negative protein, what achieves is that when it's expressed, it causes telomere uncapping, similarly to what happened during replicative stress. So we use these two models of senescence to screen for drugs that might delay the entry into senescence, or might either inhibit or delay the entry into senescence. And what we did, and this is the oncogene-induced senescence model, is, so we have here, and just as an example, proliferating cells, and in green is BRDU, and we can see that many cells are positive for BRDU. When we activate oncogene-induced senescence by adding tamoxifen that activate the RAS oncogene, so we have this senescence growth arrest, and we see that very few cells are able to proliferate and incorporate BRDU. And in this case, we treat cells with an inhibitor of TGF-beta that before we have seen that it's involved in these autocrine and paracrine loops that reinforce senescence. And we knew that if we inhibit TGF-beta, so many more cells will incorporate will incorporate VRDU and will continue proliferating. So we use this as the controls of our screening, and we screen this library. So we're screening 2 models of senescence, part of it to try to see what common things we could identify, and we identify a few drugs. And the top of those, that is this one that we call 3DA, that when we treat senescent cells with these drugs, basically we inhibit or we blunt, we rather blunt or delay the entry into senescence. And we thought that it will be good to test whether this could be relevant in some therapeutic context. And in particular, in this case, what we did is collaborate with Pura Muñoz, it was based in Barcelona, so now it's in the States, working for Altos Labs. And she has been working on basically mast cell stem cells for quite a while. And she discovered that there is as mast cell stem cells, usually they're in a quiescent state. They become activated if you have damage and you need to regenerate the tissue, the muscle. But as you become old, in what she calls geriatric mice or geriatric also human donors, so these muscle stem cells will enter into senescence, and this senescence response will stop this ability of the cells to be mobilized and to be able to regenerate muscle in response to injury. So what we thought is that if we have a drug that inhibits or delay or blunt senescence, we might be able to reestablish the regenerative potential of some of these muscle stem cells. And we did that in a setting of transplants where we have a mice that has suffered a muscle injury, and then we will transplant it with muscle stem cells to try to regenerate the muscle. And this will be coming from either young donors or geriatric mice. And these geriatric mice, they will not be able to regenerate the muscle very much. And what we did is during the ex vivo expansion of these stem cells in tissue culture, we treat them or not with 3DA, with our molecule that was able to blunt senescence. And this is what we get once that the cells got transplanted, and we wait for a while. So here in the left, so we have like the section of basically a muscle that has been transplanted with young stem cells. And we can see that there are many muscle fibers. So both in numbers and in quality, if we just look into the diameters, so there is a good number of these cells that have been able to engraft and that have been able to generate good muscle fibers that have a good diameter. When we transplant geriatric muscle stem cells, so both the numbers and also the diameter of these fibers is clearly diminished. So these don't have the quality or the ability to generate a good transplant that is going to generate good regeneration. But this is improved when we just treat. And I just want to highlight that we are treating just ex vivo during a week, during the time that we are basically expanding these stem cells. So during this time, if we treat with 3DA, so these cells will engraft much better and will basically recover the regenerative potential of these muscle stem cells by inhibiting this senescent response. So this is one of the ways that we can try to target senescence by blunting senescence induction. Of course, this has limitations on how we can use it. A good example is what I show you now. So we can use it for transplants and treat ex vivo with these drugs to improve the engraftment potential. We don't want to treat in vivo, or we treat a patient or a mouse model, because potentially I show you that senescence has some tumor suppressive roles. We don't want to need senescence in that context, because potentially we could finish with more tumors instead of having a better regeneration. Another way that we can try to target that, and I'm not going to talk about this, we published a few years ago, is by specifically inhibit the inflammatory SASP. And what we did is to basically screen for molecules that will maintain the senescence growth arrest while inhibiting the SASP inflammatory program. And what we saw is that those molecules that inhibit chronic inflammation have a positive potential to basically ablate some of the negative effects of senescence. And again, still there, you will have senescent cells that will persist. And potentially one of the problems that you have is that you need to inhibit this inflammation all the time. And another way that we can try to target senescence that we are at the moment basically reviewing for publication, and hopefully we can talk about this in more detail soon, is to try to potentiate this immune-mediated elimination of senescent cells. And what we did in this case is look for ways in which we can enhance the natural killer cells elimination of senescent cells. And these ways will eliminate senescent cells and eliminate some of the potentially negative effects that accumulation of senescent cells has. And I didn't talk much about this, but these are three ways in which we can target senescence, but without any doubt, the most popular way, and that has been explored by many, and that is tried to be used in clinical trials, and that both pharma and startups are looking, is not any of those, but basically what we call senolytics. So the small molecules that achieve the selective elimination of senescent cells. And the idea here is that if we have like negative effects associated with these senescent cells that aberrantly accumulate, if we can have drugs that selectively eliminate senescent cells, they will restore what happens normally, and we will have, again, these positive effects of senescence without having these detrimental effects associated with accumulation of senescent cells. And we call these senolytic therapies. And the idea of this selective elimination of senescent cells comes from a number of studies from the Van Dersen lab that started in the early 2010s. And what they did is to generate this mouse model that they call INCATAC, where selectively under the control of the P16 promoter, so P16 is a cyclin-dependent kinase inhibitor that is induced on senescence and is used as a marker of senescence very often. So under the control of that promoter, they put a inducible gene that is a suicide gene. So it's a drug activable version of caspase-8. So it's a version of caspase-8 that when it's expressed is non-active, but you can add this small molecule and it will dimerize. And after dimerization, will basically trigger senescence, will trigger apoptosis. So basically these mice, what they achieve in a totally artificial way is to have a selective way to kill senescent cells. And what they observe is that when they do that, the selective elimination of senescent cells extend the lifespan of mice. Not only that, but mice that have the senescent cells eliminated, they seem healthier, so they have an improved health span. So these are like almost 2-year-old mice. That one has their senescent cells eliminated, the other not. And you can see that the one that don't have senescent cells look much healthier. The extension of the lifespan is achieved through delay of cancer-related death. So that's mostly what explains this extension of the lifespan into the mice. But that's not the only thing that eliminating senescent cells does. So work that was shown in this paper and later on has shown that by eliminating senescent cells, you can have a positive impact in an increasing list of pathologies that includes, as I showed you before, many varied pathologies from cancer to fibrosis to neurological disorders to other types. And there were like two more things that make these senolytic therapies exciting. One is that you don't need to be treating continuously to eliminate senescent cells. Intermittent elimination of senescent cells is sufficient to achieve benefits until senescent cells reappear. And the other thing is that the side effects associated with senolytic therapies, and this is something that we are still fully understanding, they seem to be limited. And in these first studies, they only saw that there is a delay in wound healing while you are treating with the senolytic molecules. So with all of that in mind, there has been a lot of exciting developments and a lot of excitement in the field for the last 10, 15 years, because potentially you are able to selectively eliminate senescent cells. You can use that as therapy for many of these list of age-related pathologies where senescent cells accumulate. You can also use it as an anti-cancer therapy, and it has many benefits. So eliminating senescent cells, potentially even like reducing some of the side effects of chemo and radiotherapy that are caused by off-target induction of senescence by some of these therapies. But perhaps, and we'll revisit this a couple of times in the talk, is in the context of cancer, this one-two punch approach is what has generated more excitement. And the idea is that a lot of the normal cancer therapies, so irradiation, chemotherapy, and also some target therapies, what they act in part by inducing senescence in the tumor that basically stop the tumor proliferating. But eventually, the senescent cells that are present there, they might cause chronic inflammation. They might help the tumor to escape, and eventually there are recurrence that is caused by these senescent cells. So what it has been suggested is that we could come in what is called a one-two punch approach, first with traditional cancer therapies that will induce senescence in the tumor, and sequentially come with a senolytic drug that selectively will kill these senescent tumor cells. This will achieve more profound responses and potentially get rid of some of the side effects of chemo and radiotherapy. And perhaps a part of, obviously, these two, part of the promise of senescence therapies is that they might be used to target multimorbidities. So there are many of the diseases that we accumulate during aging in which senescent cells are present. And if we are able to eliminate these senescent cells, we might achieve benefit not just in one of these diseases that we might have on aging, but in many of those. And this might be a way to treat multimorbidities and eventually potentially might result in further benefits as shown in mouse models. So with all this potential and promise for senolytic therapies, obviously the researchers working on senescence and the pharma companies and the startups have been trying to look for drugs that achieve this effect, these senolytic drugs. And the first ones that were identified were BCL2 family inhibitors. They target the apoptotic resistance of senescent cells. So senescent cells are primed for apoptosis, but they upregulate BCL2 anti-apoptotic proteins, and therefore seems to be more susceptible to the inhibition of these BCL2 family proteins with drugs such as navitoclax, this ABT263, and others. The thing is that some of these drugs have been before used on clinical trials for leukemia and lymphoma, and they have important side effects such as thrombocytopenia and neutropenia. So potentially that might not be an issue on senolysis because we are just, the idea is just to treat intermittently with these drugs, but this highlights the need to have better drugs and a good number of drugs that can be used to target senescent cells. And this has driven research on trying to understand better the liabilities of senescent cells and identifying senolytic drugs. And our lab has been doing this for probably more than 10 years. And we have followed different approaches to try to identify new senolytics. For example, we have tried to take advantage of what we know of senescence and try to design some senolytic drugs in a hypothesis-driven way. And for example, one of the things that I mentioned before is that senescent cells have this high lysosomal activity that results in high beta-galactosidase activity. This is used as a marker of senescence. And what we thought is that if we have drugs that basically are prodrugs and that only will become active once that galactose is cleaved of these drugs, these galactose prodrugs will be more efficiently cleaved and processed on senescent cells, and potentially will selectively kill senescent cells. And we saw that, in fact, that's the case. And as a few other groups saw that if you generate some of these galactose-derived prodrugs, you will have some specificity or selectivity towards senescent cells. On top of that, what we have used is functional assays, so functional screenings, as I mentioned before, that we like to do in our lab. Like, for example, we did drug screenings in which we look for drugs that we could repurpose as senolytics, but also genetic screening, both CRISPR and RNAi to try to look for senolytic targets. And I will talk about these two in this talk. So this is some of the efforts that we did to try to identify basically drugs that we could repurpose as senolytics. So the idea that we did here is we use two models of senescence, and we basically treat these senescence or non-senescent cells with FDA-approved drugs. And this is the summary of, for example, what happened in oncogene-induced senescence. So we have the viability of the senescent cells in this axis, viability of the non-senescent cells in the other axis. Most of the drugs will fall in this cloud. That basically the drugs are doing the same to senescence and non-senescent cells, and only a few will fall in this quadrant. That will be the drugs that selectively kill senescent cells or reduce the senescent cell numbers, but do not kill or not so efficiently the non-senescent cells. And for example, and we published this a few years ago, so we found that ouabine and other cardiac glycosides such as digoxin and digitoxin are senolytics. And basically, so let me show you in this graph. So what we have here is at different concentrations of the drug. So the killing of senescent cells in red and non-senescent cells in black. And what we can see is that we have a nice window of selectivity where the drugs are killing very efficiently the senescent cells without affecting the non-senescent cells. And the other thing that I want to mention is that, so these two curves here are for digoxin and digitoxin. So these are drugs that have been used for treating patients with arrhythmia for many years, and there are hundreds of thousands of, even if they are not used so much now or not at all now. So there are hundreds of thousands and if not millions of patients that have been safely taking these drugs. So potentially they can be repurposed in the clinic. So the data that I'm going to show you is with ouabine, that is another member of this cardiac glycoside family, works same as this. And for example, this is data on all mice, so that all mice accumulate senescent cells in the liver that we can monitor with this beta-galactosidase activity or by other means. If we treat all mice intermittently with ouabine, we reduce the numbers of senescent cells. The mice will also perform better on physical tests such as Rotarod. And when we look into chronic inflammation that is revealed in this case by staining of immune populations in the liver that accumulates in all mice. So this chronic inflammation, this chronic immune infiltration basically disappear once that we eliminate these senescent cells by treating mice with ouabine. So this is in the context of aging. I'm going to show you some data in the context of cancer therapies, and I will come back to this at the end of the talk. So this is what I mentioned before, this one-two punch approach. So cancer cells, if we treat with chemotherapy or radiotherapy, we induce senescence in the tumor. If sequentially we can do with senolytic drug, we can eliminate these cells more efficiently. So this is a hepatocellular carcinoma cell line. So we treat with etoposide, that is a kind of chemotherapy, palbociclib, that is used as CDK4/6 inhibitor in breast cancer tumors, or aurora kinase inhibitor. All of these cause senescence and arrest these cancer cells, but the cancer cells are still there and potentially they can escape. If we come sequentially with ouabine, with this senolytic, the cardiac glycoside, so that doesn't make anything or much in the tumor cells per se. But when we come sequentially after treating with these senescence-inducing drugs, we can see that we can eliminate very efficiently these senescence tumors. So potentially this can be used as combination therapies. So just recapping here, senolytic drugs can be used to treat many senescence-associated diseases. Cardiac glycosides such as ouabine or digoxin are senolytics, and cardiac glycosides eliminate senescent cells in aging and cancer. And this is an example from retrospective analysis of patients that have been treated with digoxin because they have cardiac problems and later develop cancers. And what we can see is that the ones that have been treated with digoxin have better survival ratios, overall survival, than the ones that not, suggesting that perhaps this cardiac glycoside working as senolytic might be through other mechanisms might explain part of these benefits. So what I showed you there is how we can use repurposing screenings for try to identify new senolytic drugs. Obviously, there is just a limited number of drugs, and we like to explore many other things that might be senolytic. And for that, we have genetic screenings, and we have done both CRISPR and RNAi. So this is, for example, similar setup that before. So both in RAS-induced senescence and therapy-induced senescence. So we screen a druggable genome library targeting 7,000 genes. So we identify, so this is oncogene-induced senescence data, a few genes that selectively, when we knock them down, selectively kill senescent cells, and the same in the doxorubicin-induced senescence screen. In common, we found a few things. So one is BCL2 family proteins, and this is already known that we inhibit BCL2 family members. So that's senolytic. Ubiquitin, we believe that this is a liability of senescent cells that's basically are producing more proteins like such as the suspenson, and we inhibit the ubiquitin the ubiquitin systems, so they accumulate this and it can cause like prototoxic stress. And the other thing that we found is multiple components of this COP1 pathway that at that time we were unfamiliar with. What is COP1? So this is the cotamer complex 1. So this is a pathway that's involved in vesicle trafficking. And what it does is regulating the trafficking of vesicles between Golgi and ER, so retrograde transport, but also ER and Golgi, and formation of early endosome and other vesicle dynamics. So there are several structural components of these COP1 vesicles that assemble in these lattices that will basically bud from membrane structures and will be filled with cargo and will regulate transport between the different membrane structures. And there are like several of those that assemble into these lattices and for example, we'll talk about COPB1, COPG2, and a few others during this talk. A part of these basically structural components, there are also some enzymatic components that what they do is regulating the budding of these vesicles. And I'm going to talk about two of those, ARF1 and GVF1. And in particular from GVF1, one thing to note is that there are like some molecules, some small molecule inhibitors that are known to inhibit GVF1 and could be used to inhibit COP1. Just to mention, so from the genetic screens and also from the drug screens that we perform, we identify multiple, both structural like COP8, COPB2, COPG1, but also enzymatic components such as ARF1. And also in the drug screening that we did, we identified Golgi side A, so one of these inhibitors of GVF1 as potential senolytic. So there were like multiple hits suggesting that the COP1 complex is essential for maintaining the viability of senescent cells. And for example, this is an example of the results. So this is the model of oncogene-induced senescence, normal cells on top. So what we are seeing here is just DAPI stainings in the nuclei, senescent cells in the bottom. When we knock down with 3 different shRNAs against COPB2, so we see that selectively we are killing the senescent cells, but not the normal cells. And this is the quantification. So this is true for knocking down multiple COP1 subunits, COPB2, COPG1, ARF1. So inducing senescence with many types of senescence, not just with oncogene-induced senescence, but also therapy-induced senescence, irradiation, replicative senescence, many cell types, not just primary fibroblasts, but also cancer cells and epithelial cells. And overall, this suggests that this seems to be a broad vulnerability or a very common vulnerability of senescent cells. And just to complete the picture, so this is what I was showing you before. This is Golgi side A, so this drug that inhibits GBF1 and inhibits COP1 vesicle formation. It's also senolytic, so it kills the senescent cells in red here over the non-senescent cells in black or blue. And it does so by inducing apoptosis. So this is a caspase-3 assay, and we can see that Golgi side A induce this caspase activity selectively on the senescent cells. So how does mechanistically that this works? So what we can say is that senescent cells rely on having this Golgi and obviously all these this protein production apparatus to produce the SASP, so these proteins that they secrete. One thing that happens if we inhibit COP1 by either using drugs or shRNAs is that the Golgi becomes dispersed. And we can see this here. So we are staining here the trans Golgi with a green dye. So that is staining TGN46, a trans Golgi protein. So here on the top, we have like normal cells. And in the bottom, we have senescent cells. So if we just look here, so senescent cells basically have like a more active Golgi that is polarized and much bigger than in the normal cells. And when we knock down COP2, what we can see is that in the normal cells, so we still have the Golgi that is present there, but in the senescent cells, it basically become dispersed and diffuse. And this Golgi dispersal, what it means is that there's no Golgi to process adequately the proteins. There will be an intracellular accumulation of proteins, like such as proteins of the SASP. And for example, we can see here that this is intracellular staining for IL-6. Senescent cells per se are producing more IL-6 because they are producing more SASP. But when we knock down COP2, we can see that there is this accumulation of intracellular aberrant IL-6. And this aberrant accumulation of intracellular proteins will saturate the autophagy machinery and will cause ER stress. And eventually this, because it's more notorious on senescent cells, will cause this ER stress specifically on the, or selectively on the senescent cells. And as a result of that, there will be apoptosis. And we believe that's basically, this is part of the, or part of the explanation of why this is a selective vulnerability of senescent cells. Of course, one thing that we want to do is to try to see whether we can apply this to treat different pathologies. And we talked that we have Golgi side A and Brefaldin A, two small molecules that we could use to try to target senescence. And the problem is that these molecules, they work very, very nicely in cell culture, but they have very poor pharmacological properties. So they can only stay in blood for matter of minutes, and then they become degraded. So they are not really molecules that we could use for pharmacological studies. The good news is that this ARF1, the protein that controls this formation of the COP1 vesicle, is myristoylated. So you have this lipid modification that is needed for their function. And we have very good NMT inhibitors. So NMT is the enzyme that is involved in myristoylating ARF and many other proteins. And we thought that these NMT inhibitors might be able to basically phenocopy this inhibition of the COP1 pathway. So this is just some proteomic studies showing that some of the top things that are myristoylated on senescent cells are these ARF proteins. And basically what we thought is that if NMT inhibitors inhibit the myristylation of ARF, it is known that that might trigger the degradation of ARF because it's not myristoylated in the N-terminus. And eventually that will mimic what happened when we knock down or degrade some of the other COP1 components, so will cause Golgi dispersal, intracellular accumulation of the SASP, ER stress, and dysfunctional autophagy. In fact, that's the case. So if we treat with two different NMT inhibitors, so the levels of ARF decrease independently of senescence, but eventually that results in this Golgi dispersal and all the rest of other phenotypes associated with this COP1 inhibition. So COP1, so NMT inhibitors are they good senolytics. So as a comparison, this is digoxin that I showed you before, this cardiac glycoside. We call it this senolytic index that is basically the difference between the EC50s in senescence and non-senescent cells. And basically this is 20 times. And this works in the nanomolar range. What I'm going to show you is, so this is an NMT inhibitor that you can buy from different providers. And this has a senolytic index of 5, and it works in the micromolar range. This is, we team with this company that is called Mirix, and this is one of the inhibitors that they have had in development. So it has senolytic index that is similar to digoxin, that is one of the best that we saw, and it's again working in the nanomolar range. And I will show you some studies using these inhibitors. So basically these ones that are here, but just to show you, this is one of the last second generation inhibitors developed at this company. And this is the selectivity between, in this case, senescent cells are in yellow and non-senescent cells in blue. This is working in the subnanomolar range and has a selectivity of millions of times. So potentially, these are very good senolytic inhibitors. I will go very quickly through some of the results that we have. So we have tested this in models of aging, senescence, cancer and disease. So this is, for example, in aging, where we are treating like all mice, so 2-year-old mice, and intermittently with NMT inhibitors. So we can see that we decrease the presence of senescent cells in the liver and the lung, and we decrease senescence-associated phenotype, like, for example, liver steatosis, so accumulation of fat in the liver that happens during age, or lung fibrosis are decreased once that we eliminate these senescent cells. I'm going to just very quickly go through some of the phenotypes that we can see on this model of high-fat diet that mimics what happened during non-alcoholic steatosis. So mice gain weight. They also have like higher levels of cholesterol in blood. And we can detect these ALT and ALP enzymes that tell us about liver damage. And these phenotypes are like partially reverted when we treat with NMT inhibitors. We also, when we have mice in this high-fat diet, they accumulate senescent cells. We revert, or we can basically kill these senescent cells treating with NMT inhibitors, and we can monitor that by immunohistochemistry against P21 or by following the transcriptome of these cells. And on parallel of that, I will just show you, like, for example, there is a decrease on this accumulation of fat, decrease of this steatosis that happens in this high-fat diet model, a decrease of the immune infiltration that happens in this high-fat diet model when we treat with NMT inhibitors, and also a decrease of the fibrosis that we see in the liver that is associated with this high-fat diet. So in general, what we can show from this is that functional screenings identify COP1 as a novel vulnerability of senescent cells. It works by triggering a SAS-dependent unfolded protein response and ER stress response on the senescent cells, and treatment with NMT inhibitors phenocopy these effects and can be used to treat a broader spectrum of senescence-associated diseases. And what I'm going to show you in the last slide of this talk is whether we can translate these senotherapies to the clinic. And I'm just going to skip this and show you an example of something that has been done by one of our collaborators, Lars Sender. And one of the things that we need, apart of having good molecules that we can use to try to target senescent cells, is good ways to monitor senescence. So they develop a PET probe that is able to basically report for beta-galactosidase activity. And this is in mouse models where you have senescent cells. And you can use this PET probe to monitor senescence. So in the context of some clinical trials, what Lars did is he had a patient with glioblastoma, very aggressive brain tumor that was subjected to surgery and irradiation. So that's make an improvement in the patient for a number of months, but eventually these tumors are lethal, they reappear. And when the tumor reappear, so Lars took the patient to the clinic, and apart of just monitor for the tumor itself, use this senescence PET to try to see whether there was a senescence signal. And indeed there was, where the tumor was, there was like this strong senescence signal. This is not a surprise because irradiation is not to cause senescence. So basically Lars maintained this patient with target therapy, but also give the patient digoxin, so as a senolytic, same regime that is given to patients with cardiac arrhythmia. And after 3 weeks of treatment, the patient had improved, came back to the clinic, the tumor had reduced in size, and also we could see that the senescence have disappeared or partially reduced in size. Eventually, I have to say this is just one patient and the tumor reappears. So glioblastoma is a very aggressive tumor. But what this showed is that if we have a combination of good ways to target senescent cells by senolytics, for example, and good ways to try to detect senescence, for example, by PET probes. And I didn't talk today about other ways that we can use to try to detect senescent cells, such as machine learning algorithms. We can try to bring these xenotherapies to the clinic. And just to finish, so this is some of our funders and some of the people that has been involved in the work in our lab and our collaborators. So thank you.
Thank you for a very interesting presentation. There are some questions, so I'll start off with the first question from Larry Parnell. And he thanks you for your informative presentation and asks, regarding the testing of agents that could blunt senescence in muscle, did you observe any measurable effects with natural or food-based compounds?
So that's a good question. So we did not look for that. So we screen for that particular piece of work. We screen like basically a FDA approved drug library. So we did not screen natural or food-based compounds, but I will assume that there will be effects of some of those.
Second question is from Chris Wiley, who says, wonderful talk. Um, with so many therapeutic opportunities, we love following your work in our lab. Do you have any concerns that targeting COP1 may have off-target lethality in vivo for other highly secretory cell types, such as immune cells or some endocrine cells?
That's a very good question, and I'm sorry that I didn't go through that in my talk. So obviously, what the phenotype that we see with COP1 inhibition or NMT inhibition is related to affecting the Golgi and affecting secretion. And there is a selectivity there towards senescent cells, but there are other cells that are highly secretory. So in particularly with NMT inhibitors, so we tested what was happening in immune cells when we give the NMT inhibitors to a high dose for 5 or more days. And we also test, for example, what was happening with secretion of insulin and some other cells that are highly secretory. We did not have some. What I should say is that these inhibitors, of course, they are toxic if you give them to a high enough concentration and for a prolonged period of time. And for example, they are being tested in clinical trials against cancer. And I think that for some of these cancer therapy purposes, so you give it to much higher doses and for a more prolonged time than during senescence. So basically, what I can tell you is that for having toxicity associated with these compounds, you have to treat to high concentrations for more than 6 days. Typically, in our studies, we were using concentrations that were like 5 or 10 times over that limit, and we were treating just for 3 days and then doing intermittent treatment. So on those circumstances, we look for immune cells. We look for example, for secretion of insulin and secretion of other factors. We didn't see any toxicity. For sure, if you treat for longer time or higher doses, you're going to achieve this. You're going to have toxicity. These are genes that are cataloged as essentially knockouts. Yeah.
Thank you. Then the next question comes from Jean-Dan Wong, who thanks you so much for your excellent work and great presentation, in particular on the paradoxical effects of senescent cells. And then goes on to say, you have convincingly demonstrated that selective elimination of senescent cells can ameliorate, even revert the pathological manifestations of multiple disorders in your mechanistic studies in vivo and in vitro. So considering the cellular senescence is context dependent, for example, aging versus cancer or risk versus benefit effect, do you know if there are any human clinical trials to prove proof of principle of the impact of xenotherapies on senolytic agents, for example, NMT combinations for multiple targets on age-related disorders and aging? Okay.
Yeah, so senolytics is a relatively young field, so but still there are a few clinical trials and first-in-human studies that are in progress. So I think first trials probably started 2016 or so, and some of the most active guys on conducting clinical trials, or at least first-in-human studies, have been in the Mayo Clinic. There have been multiple studies using mostly dasatinib and quercetin or fisetin that are dasatinib is a tyrosine kinase inhibitor, but fisetin and quercetin are natural compounds, and they have been shown that they have senolytic properties, and there have been multiple clinical trials started there on different diseases And yeah, I think that I will say that last time that I look, probably there are like 30 or 40, most of them small investigator-initiated trials. There are some companies that are also conducting some of these trials, and Unity was one of the companies that started and had basically a senolytic trial in osteoarthritis that basically failed on phase 2. But now they have like some trials with BCL2 inhibitors on ophthalmological disease that have had a very successful phase 2, and they are just progressing to phase 2b. So I think that there are some trials happening.
Okay, and the next and maybe last question comes from Kriska Kuhneman, who asks, do CGs and NMT inhibitors show a similar extent of senolytic activity in multiple models of senescence induction other than oncogenic activation?
The answer is yes for these two. So we selected these two because precisely because of that, because they have what we call a broad spectrum senolytic activity. That what signal is that they are targeting some pathways that seems to be important for many types of senescence. But there are also many, and of course the devil is in the detail. There are many senolytics that are specific of a specific cell type or a specific mode of induction of senescence, and we have seen many of those, and perhaps for using them in some specific context, this will be more relevant because potentially they may have a more restricted effect and less side effects.
We do have one more question here from Larissa Constantino, and she asks, are the doses of digoxin as senolytic, are they the same as for treating heart disease?
Yeah, so we look into that, and I don't remember the numbers now, but basically, I think that the concentration that you can achieve in blood of digoxin or digitoxin are in the low nanomolar, and what we could see is that in similar concentrations, and I'm talking from the top of my head right now, but I don't know if it was like 50 nanomolar or between 50 and 200 nanomolar, we could achieve senolytic effect, at least in some cells in cell culture. So we thought that we were in the same range that is achieved in patients, and that has been reported that you can achieve in the blood of patients. So, yeah.
Okay. And I see no more questions. So again, I'd like to thank you for a very fascinating presentation.
Thank you very much. Thank you, everyone. Bye-bye.
Thank you, Dr. Hill. Okay. Bye, Paul. Bye,
Bye.
Peggy.